The first lecture that I'm going to give you today is a little bit of an introductory lecture. I want to go over some of the concepts that you should have in your mind, solidify some of the ideas that you should have got from previous neurophysiology, from anatomy, from different areas of the course, and put them in a physiological context. And there'll be quite a lot of that throughout the paper this semester, taking things that we hope you've grasped by now and putting them into a formal physiological context so that you can think with them in a physiological way and use them in a clinical way to solve problems, which is where you'll be going in the next couple of years. So, functions of the central nervous system. It's a squidgy grey box in your heads. What does it do? What's it for? Everything. Digests food, transfer. Well, no, I hear what you're saying. It doesn't do everything, but it does allow everything else to do its thing. Yeah? It's reasonably important, at least in a mammalian context, to allow us as mammals, and most of the animals that we will deal with as mammals, or if not mammals, at least vertebrates, it allows those animals to function within their environment and to survive and reproduce in their environment in a way which is conducive to their DNA getting to the next generation. That's what it's about. And so it integrates all the other functions, and that's why we're dealing with it almost last. Divisions of the central nervous system. This is the mammalian central nervous system, and I want to emphasize that again and again. When you look at the central nervous systems of the other vertebrates, they are very different from the mammalian central nervous system. And it's hard enough to give you an understanding of the mammalian central nervous system. We're not going lower down. But please remember, if you do deal with a patient that's a bird or a reptile or an amphibian, it doesn't have a central nervous system that's the same as a mammal's. It's similar, but some of the bits are different. And some of the functions of the bits that are different are different. So, we start off with the spinal cord. And the spinal cord will be the subject of the next lecture. And then we go up through the medulla, the pons, the cerebellum on the side, the mesencephalon, the diencephalon, and the cerebrum. And all those areas are just words and boxes. One of the things that I want to emphasize throughout the course is the interaction between the anatomy and the physiology in the central nervous system. Of all the physiological systems, the central nervous system is the most anatomically dependent. I think we talked about that at the start of the 204 lectures, where I showed you a slide of a neuron and a slide of a hepatocyte. You guys remember that? And I said hepatocytes don't really mind where they are within reason. As long as they've got a blood supply and somewhere to dump the bile, they can get on with being hepatocytes and everything's happy. A neuron, particularly a neuron in the central nervous system, is entirely dependent on its anatomical shape and configuration and location to perform the function that it performs. And we saw that in vision and in hearing last semester, where we looked at the locations of neurons in their nuclei and said, well, this nucleus is doing this, receiving inputs from here, sending outputs to here, and it's all about the way that it's wired in, it's all about its location. Neuroanatomists talk about a 2 millimeter scale within the central nervous system. They say that within the central nervous system, if you're looking at a neuron and you understand what it's doing, and you move 2 millimeters in any direction, you can be dealing with a neuron that is completely different in terms of structure, function. Everything about that neuron can be completely different on a 2 millimeter scale. That's interesting for two reasons. One, because it gives us an extremely complex machine. Yep, the mammalian central nervous system is the most complex machine in the whole universe. And it has been said about it that if it was simple enough to understand, we would be too simple to be able to understand it. Does that make sense? Yeah? Because we have such a complex thinking machine, we can actually approach it and look at that machine and try and decide what it's doing. But it's so complex, giving us all those functions, that we can never actually get into it and understand it. So what I'm going to give you is a pasty overview, which hopefully will give you enough information for an understanding at a clinical level. And what I'm hoping you'll all be able to do by the end of the course in neurophysiology is look at an animal with a neurophysiological lesion and be able to say to yourselves, I can work out where that animal's lesion is based on the clinical signs, because I know what the dysfunction is looking like, I know which systems that dysfunction is being generated by, I know where they are, and I can hone down on where that problem is anatomically, because I know enough neurophysiology to be able to do that. And so in a spirit of linking the anatomy and the physiology, here is half a dog's brain. I've sliced it down the middle, and I'm just going to highlight the different areas for you at a very basic level to get you into the idea of where the different bits are and what they do. And the other concept that I want to get into your mind is that the central nervous system is, to some extent, not wholly, but to some extent, and certainly in a clinical consideration, it's a bag of progressively more complex reflexes. So we'll see tomorrow that down here in the spinal cord, we have reflexes that link sensory and motor function. And up here in the medulla, we have another set of structures that link sensory and motor function related to the autonomic nervous system, and you'll have met those already. The cardiovascular center, the respiratory center, you guys met those in physiology? Yeah, you remember that they live in the medulla? Yeah, so they are a set of interactions between sensory and autonomic motor that live in the medulla. The medulla is also responsible for most, but not all, of the cranial nerve sensation. Certainly most of the cranial nerves that carry somatic sensory information get in through the medulla. Next up is the pons. Pons is Latin for bridge. The current Italian is ponte, like the Ponte Vecchio. Yep, it's a bridge. And it bridges not only motor function which it integrates, but also the two sides. It's one of the main bridges between left and right. So, in the ventral pons, this area here, we have fibres which cross the midline. They girdle the ascending and descending fibres, and they allow the two halves of the cerebellum to communicate with each other. And they integrate motor function across different areas. The cerebellum, more Latin, little brain. But don't let its little brain name fool you. It contains something like 40% of the neurons of the central nervous system. Okay? In terms of thinking and in planning motor actions, as we'll see later on in the course, it's a big mover. It knows what's going on. It doesn't always do much about it, but it knows what's going on. It can affect motor function. It can think about motor function, plan motor function. It can initiate motor function. The mesencephalon, midbrain, is a coordinator of sensory and motor function. It's all about making motor function more basic and more relevant to the muscles. And it's all about coordinating sensory function and to some extent sensory feedback with that motor function. And now we're getting into the higher centers. The diencephalon, forebrain, is responsible for autonomic and endocrine function, the hypothalamus, the bit under the thalamus. And you'll remember when we covered that last semester, we're not talking now about relatively simple autonomic feedback, cardiovascular respiratory function. We're talking about more integrated autonomic feedback. Thirst, hunger, reproductive function, life cycle function, all of those complex autonomic functions that keep us moving through our lives at the various different stages are processed and sorted out within the diencephalon. And in the structure of the thalamus, or I should say the thalami, because there's one on each side, processing of information to the cerebral cortex. I used to be able to say that no information got to the cerebral cortex without going through a synapse in the thalamus. It's not quite that simple anymore. Some information does get to the cerebral cortex without going through the thalamus, but not very much. The thalamus is still the last arbiter of sensory information going up to the cerebral cortex. And linking in a circuit with the cerebral cortex, it's a big arbiter in control of the cerebral cortex, in conscious thought, in arousal, in processing information, dealing with that information, and allowing that information to affect the animal's brain as a whole. The corpus callosum, rigid body, is the last of the big connectors between the left and the right brain, and in this case between the left and the right cerebral hemispheres. Myelinated axons, something like 6 billion of them in each of your brains. Huge numbers of fibers connecting between the left and the right brain. And last but by no means least, or at least not for us, the cerebral cortex. Responsible, at least in man and the primates, for fine motor function. We will debate that in a few lectures' time. Responsible largely for memory. Responsible largely for emotion. The thinking man's brain, as it were. So those are the areas of the brain. As we go through, we're going to be looking in progressively more and more detail at what I call the grey blobs, but looking at different structures within those areas that communicate with each other. And at least in giving you those areas, I hope I've given you an anchor to pin things on. So that if we say, oh, I don't know, the cardiovascular centre is in the medulla, you'll be able to say, oh yeah, about there. Yeah, it's pin the tail on the donkey time. Think about where the structures are, and it gives you a clue as to both what they're doing and also how they relate to each other. The principles of organisation are pretty much these, and we have met them all, I think, pretty much. Each system, by which I mean system of touch, system of heat, system of vision, system of hearing, system of pain, system of olfaction, all the systems involve several regions that carry out different types of information processing. There are identifiable pathways that link the components of a system so that we can look at a white matter tract. The white matter tracts tend to be axonal because they're myelinated, and myelin is a fatty substance, so it's white, as opposed to the grey matter nuclei, which tend to be cell bodies. So when we're looking at a white structure, like the fornix last semester, we can say that goes from there to there, and it's an information superhighway, it's a linker. So those pathways that link the components of the system, we can see them. We can touch them and prod them and say, there they are. The pathways project in an orderly fashion. It's not a mishmash, it's not just thrown together. It's all made so that things smoothly align with each other at every level. Systems branch out, systems branch in, but it's all very orderly and looks almost nothing like an Auckland street map. Okay? It's all put together in a sensible way. Systems are hierarchical, and we met that particularly in vision and hearing last semester, where we were talking about information processing going on at different levels. And lastly, systems decussate. Almost all of the neurological systems decussate, by which I mean they cross the midline. So that for the most part, and you'll remember vision, this side of the body is dealt with by this side of the brain. Yeah? Different systems decussate in different places. Some systems decussate and then decussate back. But all but one system that I can think of off the top of my head decussates at some point, crosses the midline at some point. And the point that I want to make as a global point, which I may have alluded to last semester, but I will say it again explicitly, is that the whole of the central nervous system is the thinking engine. The whole thing is what does the thinking. There is a very sort of schoolboy view of the brain, which is promoted a lot in popular science magazines and programs, that suggests or implies, if not suggests, that sensory information comes into the spinal cord, is taken up to the cerebral cortex. The cerebral cortex goes, we'll do this. And then the motor information goes back down the spinal cord and goes out to the structures, the muscles, to do something. Yeah? Now, if you functioned along those lines, your spinal cord would need to be about as thick as your thigh. And your brain would have to be about as big as a telephone box, a proper English red telephone box, you know, not one of these nasty little stand and try and huddle and talk on it, a proper big telephone box. And imagine a system this big takes 20% of your cardiac output, okay? Make it as big as a telephone box. How much cardiac output does it need now to keep going? And how are you going to carry that up and down trees and along and farmland and all the sorts of things that your ancestors have done to get you here? Is that going to happen? Probably not, okay? The point I'm labouring is that information crossover and decision making goes on at every point, from the reflex that Gordon talked to you about right up to the cerebral cortex. And the different layers make decisions about that information at an appropriate level. It's a bit like a computer, in that overall the computer has a function, or a series of functions. You know, it can entertain us while we shoot aliens or do Grand Theft Auto or whatever we do in an evening. It can get us an education while we type away at an assignment and get it printed out looking nice with colour pictures. It can order something from the latest Amazon bookstore or the record shop. It can download music. It can do specific things. Different parts of the computer do specific functions, but there isn't any one part of the computer that deals with typing of assignments and another part of the computer that deals with ordering from Amazon. Okay? The different parts of the computer work in unison with each other, work together to produce those functions. And so when you look at a computer, it's kind of inappropriate to say, well, that bit does that, and that bit does that, and that bit does that. You lose something if you look at it at that level. You need to keep the whole of the functionality in your mind, or rather the whole of the system in your mind when you're looking at functionality. So when you're dealing with the central nervous system, this is really hard at this stage because you don't really know what's going on in different bits. And after 20 years, neither do I, but that's another story. When you're dealing with the central nervous system, try and deal with it in a holistic manner. Okay? Try and think about a specific function and think about how is the central nervous system as a whole working together to provide that function. And I'll give you examples of that throughout the paper, but I'm really interested in that overview, because that overview is what's going to give you the clinical insight to be able to diagnose problems in patients. Oh, some physiology at last. This is a basic view of a nervous system. There is an integration or processing center, usually illustrated at the top, because most textbooks are very homocentric. They're very people-oriented, and so the thinking centre's always at the top. If you're going to get veterinary, you can try and put it at the left or the right or wherever you want to put it, but the middle of the spine always gets in the way and makes for messiness. But somewhere there is an integration processing centre. The processing centre generates a motor output. I will go to the pub and a motor output. There is feedback from the motor sensor to the sensory sensor. This is not the way to the pub. And the integrating centre works with that sensory information to modify the motor output. Now, of course it's not. I should have turned right 2 streets ago. Back we go. Okay? It's all about deciding what you want to do, affecting a motor output, using sensory information to decide if that motor output was adequate or effective or sufficient, and altering the motor output in order to deal with that. When nervous systems do that, they have a whole string of features, physiological things that they do, ways that they do it, features that they show. And I want to spend the rest of this lecture talking about those different features. And we're going to call them properties of an integrating centre. Now, an integrating centre can be something quite small, like a lower motor neuron in the spinal cord, a single neuron, or it can be something quite large, like a cerebral cortex. But when I use the term integrating centre, I'm meaning a physiological structure that is involved with processing information in some way. And these are the things that those centres do. And so what I want to do now is to go through each one of these in a physiological sense and talk about them just to cement what they mean and what they do in your minds. You will have met most of them, you might have met all of them. And where I've told you about them before, I'll be using the examples that we've worked with previously to go through them, just to cement them in a physiological sense. Convergence, first of all. Convergence is the concept of many axonal or neurological pathways converging down into a smaller number of pathways, usually down to a single pathway. This black-stained neuron, with its cell body here and its dendritic processes here, is a lower motor neuron. It lives in the spinal cord. We'll meet it in a lot more detail in the next lecture. But for the moment, I want to use this neuron as an illustration of a convergent pathway. This dendritic tree takes synapses from upper motor neurons, from central nervous system neurons, which control or contribute to its function. A single lower motor neuron can have around about 50,000 synapses from other neurons on its dendritic tree. And each one of those 50,000 synapses can contribute to the usually single axonal pathway that comes off that lower motor neuron. And that single axon goes down to a particular muscle group, to particular fibers within that muscle group, and causes them to contract. So it's an integrator of all of these processes which are influencing it. Some of them might be reflexes from sensory neurons within the same segment, or long reflexes from neurons within other segments of the spinal cord, or neurons coming down from higher centers of the central nervous system. This thing integrates all that function and is convergent in the sense that all those possibilities come down to one axon and cause the contraction of one small set of muscle fibers. And one of the hallmarks of motor pathways is that they are convergent. Motor pathways almost always convergent. They take information from all over the place and they converge down to a single output pathway. The opposite of that is divergence, a single sensory input pathway that diverges out to a whole lot of other pathways. One of the places we saw that last semester was in the optic pathways, and hopefully you'll remember this diagram. Everybody nods wisely and sagely because they learnt it for their exams. Good on you. And you will notice that the optic nerve is coming in to the lateral geniculate nucleus and the rostral colliculus and diverging, sending information to the visual cortex, to the reticular formation, to the cerebellar cortex and the tectospinal tract, and to the motor nuclei and the parasympathetic nucleus responsible for controlling the eye. It's a sensory system, it's divergent, and the hallmark of sensory systems is that they are divergent. They take sensory information and they spread it to lots of different locations throughout the central nervous system where it's going to be useful. Delay is a consequence of neuromuscular function. This is the electromyogram of a quadriceps femoris muscle after the patellar ligament was stimulated at time 0. It's a very particular electromyogram in that it's the electromyogram of a professor of surgery. But for all of that, I think it's probably the same as the EMG of most normal humans. You can see that when you stimulate the patellar ligament, there is no electrical activity in that quadriceps muscle for 24 milliseconds in this case. And then suddenly there is a big compound action potential and contraction which takes place in that muscle sometime later. And that delay is a consequence of the time it takes the sensory information to get up to the spinal cord to process that reflex with its synapse, and the motor information to get back down from the spinal cord to the muscle and to cross the neuromuscular junction, and the last little bit of delay between electrical depolarization and mechanical contraction of the muscle. And you should by now be able to follow all the processes in that delay. You know about the generation of an action potential from a depolarization, a change in membrane potential. You know about the speed of action potential propagation as the different channels open and close, and usually in a myelinated pathway you know about the saltatory component of that, which speeds things up. You know about how synapses convert electrical energy to chemical energy and then back to electrical energy to cross the gap between cells. And you know about the way that muscles contract and the way that calcium influx causes a contraction of the muscle fibers sometime after the electrical activity. So all of those delays add up to 24 milliseconds. But delay is a consequence of the way nervous systems work. You can't have processing of information, well, you can't have processing of information of any kind without delay. But particularly in the nervous system, we see that delay and have to account for it. One of the consequences of delay is that it tends to lead to long neurons. Kind of debating as to whether or not I was going to get into this, but I will spend a couple of minutes on it because it's quite interesting. If you have a long neuron, huge, great long thing, as long as you can imagine, then that is the fastest way to get information from this point to that point. Because we don't have any synaptic delays, the only delay we've got is the delays the action potential travels. It doesn't stop at any point to do the electrical conversion. The consequence of that long neuron is that it's extremely metabolically costly to keep a cell going that is, well, in a horse it might be 3 meters long. Yeah, a 3 meter long cell is a ridiculously difficult thing to keep alive in a metabolic sense. And it's also quite delicate. If you've got a 3 meter long cell that is this thick, how easy is it going to be to damage that thing? Very easy. And so there's a balance between the metabolic cost of these cells and the fragility of these cells and the speed of information processing. And the central nervous system is a fine balance between lots of short fibres and a few long fibres in particular places which allow information to jump into different places. So fibre length is quite critical. And the reason it's critical is because of this delay in transmitting information. We met fatigue, again, in the visual system. You'll remember we said that if we shine light on an area of the retina, we see a change in the rate of action potential generation in the nerve fibres of the optic nerve, the appropriate nerve fibres. And we said that if we shine a light and we keep that light shining constantly, we get a change of rate and then it fatigues back to close to its original rate. People remember that? Fatigue is a consequence of nervous function, or consequence of. No, fatigue is it's a given, really. It's a parameter of nervous function. And so when we process information, we process it in a way that understands that fatigue and codes for the speed of that fatigue. And again, there's always an exception that proves the rule, and we'll meet it later in the course, but I can only think of one neurological system that doesn't undergo fatigue. Redundancy and lateral inhibition kind of go together. Redundancy is a kind of a flexibility, not doing anything, extra function type thing. And you'll have met redundancy in all of the physiology that you've done so far, although you might not have heard it called redundancy. Jeremy is just there to remind me to talk about redundancy. I'm not going to bother you with why. In terms of cardiovascular function, last semester we met a sheep that could lose a certain amount of its blood volume without getting into physiological trouble. Yeah, about 10% of its blood volume, not a big deal. Spare capacity, redundancy. Yeah. In terms of respiratory function, most people can get by more or less well enough on a single lung. I don't recommend you go and see a cardiothoracic surgeon and test that out, but if you needed to, you could probably get by quite well on a single lung. Redundancy, spare capacity. Most people would only need a third of their liver, assuming that they didn't have a history of being vet students and so didn't have chronic cirrhosis. But for most people, a third of your liver is what you need. And it's also true in the central nervous system that there is spare capacity or redundancy. But the nervous system is so attuned to its function that redundancy tends to get used in different ways. And we tend to think of it as the concept of, if this pathway is damaged, there are other pathways which can take over that function. It might take them some time to do so. They might never be as good at that function as the original pathways were, but there is spare capacity in the central nervous system at all levels to take over function that's lost, up to a certain degree, a degree of redundancy. Feedback is a feature of motor systems. I've shown you that already, that a motor system causes an activity, feeds back to a sensory system, and that alters the motor activity. There are three kinds of feedback, and the simplest to understand is a ballistic control system. This is a UK archer from the Olympics about 8 years ago. She's a particular hero of mine for the simple reason that she can take that arrow and put it into a space that big, 120 meters away, which is going some, really. In fact, when she's shooting in competition, because the arrows twang when they hit the target, she has to put the arrows in different points of the target. She never aims for the middle, because if she aims for the middle twice, the arrows twang against each other and damage each other, and she has to get new ones. The point is that she's doing a fairly complex motor function. loosing an arrow. Once the arrow's gone, she can have no influence over the course of the arrow. It's gone and it's on its way. And once she gets feedback, there it was on the target, there's no way of changing the motor function to change where the arrow went. So it's ballistic in the sense that you throw it, you see what happened, and then you can throw it in a slightly different way next time to adjust the result. So we have a desired result. The arrow goes in the middle, I get a gold medal. A series of stored programs. If I move my arms in this way and do all this business, then the arrow goes into the middle, I get the gold medal. We have a controller, a central nervous system, which generates a motor output based on the desired result and the stored programs. We have a contribution of noise. The wind was blowing that day, it was gusty, it was moving in this direction, and we have an actual result. And then the actual result will come back here and adjust the stored program slightly so that next time the archer tries to get the desired result, the controller will be slightly different. It's learning by experience in a suck it and see sort of a way. Try it, didn't work, try it again, bit better, try it again. That's where we want it to be. The other sort of motor feedback is a continuous feedback, where the actual result can be fed back by a comparator into the controller in real time. And we see that in gymnastics. This gymnast on the high rings is performing a crucifix, a very difficult move, which involves kind of rolling over and twisting all your arm muscles up. But he's getting feedback from his arm muscles at every stage of the process. There's a desired result. I want to do this gymnastic maneuver. And the controller is taking information back from the body the whole time and adjusting the motor output continually to make the actual result match the desired result. There's still noise there, but this time it's dealt with all the time, on the fly, instead of in chunks. So those are two of the three sorts of feedback. The last sort of feedback is a bit of a cheat. It's feed forward. And it's when you're catching a ball or not catching a ball. And I love this slide because when I was growing up, it was quite unusual to see a West Indian backstop not actually catch a ball. But it's sadly more common these days. When you're catching a ball, you're working out where the ball is going to be at some future point in space. And you're working out how to get your body and your hands into the right position to intercept that ball and bring it in and catch it with nice soft hands into the middle of your chest. So feed forward is feedback in a predictive sense, in a future sense. Facilitation we owe to Charles Sherrington, Sir Charles Sherrington, who won the Nobel Prize in 1932 for his work and is acknowledged as the grandfather of modern neurophysiology. And the reason he's acknowledged as the grandfather is because before Sherrington, there were people like Golgi who could stain neurons, like that really nice silver stain that we saw a few slides ago. That was a Golgi silver stain, and could say, that's a really nice neuron. Wonder what it does. And people like Pavlov, who we'll meet in a minute. Remember Pavlov's dogs? Yeah? Pavlov had some really profound things to say about the way the central nervous system carried out learning, but didn't actually know which bits of the central nervous system carried out that learning, and didn't have the tools to be able to ask that question. He was just saying, central nervous systems learn like this. Sherrington, for the first time, was able to say, neurons work like this, and it's those neurons doing it. He was the first person to tie together the physiology and the anatomy at a cellular level. And his concept of facilitation involved these relatively difficult to interpret diagrams, but these are the originals. He said that if a sensory pathway comes into a neural system, it causes depolarization of some neurons. But there are other neurons around the edge where it causes a change in membrane potential that doesn't reach threshold, and it facilitates those. And if you can imagine, system A comes in and fires its set of neurons, and system B comes in and fires its set of neurons, there might be neurons that are facilitated by both A and B, but not taken to action potential by either A or B. But if A or B come along together, and you get A and B, then those facilitated neurons are doubly facilitated, and they reach action potential. So he was saying the output from A and B together is greater than the sums of the outputs from A on its own and B on its own, and that's facilitation. It's the concept that a sensory system can alter the function of neurons, not sufficiently to bring them to action potential, but to alter their membrane potential so that it's easier for a second system to bring them to an action potential. Summation you'll have met. It can be temporal or spatial, and you'll have met it in terms of inhibitory and excitatory postsynaptic potentials with Gordon. Yep, everybody happy with those? Don't need to go into those in any great detail. Nobody objecting. Inhibition is the system whereby an inhibitory neuron can influence or turn off an excitatory pathway. And inhibition is hugely important in the central nervous system. If you think about it, if all of your neurons were excitatory and only excitatory, then a sensory input in any area would ultimately lead to all of your neurons firing, because the first neuron excites the second neuron, which excites another couple, which excites some more. Everything gets very excited, and you go, oh, can't do anything, okay? It's the inhibitory neurons that do the thinking. They are the ones that turn on specific pathways or allow specific pathways to be turned on, turn off other pathways, can turn on or turn off parts of the dendritic processes of individual neurons and alter the way that they process information. A simple example of that is reverberation. Here is an excitatory pathway in red down the bottom that goes round and round and round and round and gets more and more and more excited until it either runs out of ATP and curdles itself into a heap in the corner, or is turned off by an inhibitory pathway. And we see those pathways particularly in the pain pathway areas, where a seemingly small and short stimulus can have a really big motor effect. Talked about memory. I'm hoping that you'll all be familiar with Pavlov and his dogs that learnt to salivate in response to a bell. Lastly, I want to talk about somatotopic mapping. Somatotopic mapping is the concept that areas in the nervous system, and particularly the central nervous system, correspond to areas in the body. We met that in the cochlea, where I said that areas of the cochlea are represented in areas of the primary auditory cortex. And you have a frequency response along the cochlea, and similarly a frequency response along the primary auditory cortex. Here we have the somatotopic map of the sensory cortex of a human. Apologies, it's the motor cortex. In Stomale days, the motor cortex of a human. The difference between the sensory and the motor cortex is in the genitals. And I'm not going to go into it in more detail than that, but if you see a somatotopic map, look at the genitals and you can work out whether it's sensory or motor. The idea is that the bigger the area, the more neurons are coding for information in that area. And you can see that areas that link up on the brain also link up through the body, so that the toes are next to the ankle, is next to the knee, and so on all the way along. The other thing to say is that we can see somatotopic mapping at almost all levels within the central nervous system. So here is the cervical spinal column of a rat, and you can see that across the sensory pathways within that cervical spinal column, the information is ordered in the same way as the body is ordered. Here we are in the thalamic nuclei of the same rat, and you can see again that the information, there are two representations now, but the information is ordered, so that through each structure in the central nervous system you can cut a slice through that structure and you can see the order of the body in the areas that those neurons deal with, in both the sensory and the motor systems. That's the rat's cerebral cortex, and that's the end of the lecture.